All right. It looks like we are at the top of the hour, 12 noon exactly. I see we still have some attendees joining in, but I want to be respectful of everyone's time and our speakers' time. So we will jump right in. So I just want to thank everyone for joining today's clinical pharmacology lecture series. It's honestly my distinct pleasure to introduce our speaker, Dr. Mansoor Khan, whose expertise and insights continue to advance our understanding of 3D printing and bioprinting. Dr. Khan served the U.S. Food and Drug Administration, or FDA, for over 11 years as the Director of Product Quality Research and a Senior Biomedical Research Scientist in the Center for Drug Evaluation and Research, otherwise known as CDER. There, he helped develop regulatory policies for reviews and compliance and led CMC review and research teams on drug delivery systems, product stability, biotech products, and biopharmaceutics. In 2015, he joined Texas A&M University as professor and vice dean at the Rangel College of Pharmacy. Prior to joining the FDA in 2004, Dr. Khan was a professor of pharmaceutics and director of graduate program in the School of Pharmacy at Texas Tech University Health Science. He earned his Ph.D. degree in industrial pharmacy from the St. John's University School of Pharmacy in New York in 1992. He has published over 335 peer-reviewed manuscripts, 5 texts, including Quality by Design for Biopharmaceutical Drug Product Development, 25 book chapters, 200 poster presentations, and more than 250 invited presentations worldwide. Dr. Khan has held several leadership positions at the American Association of Pharmaceutical Scientists, including elected chair of pharmaceutics and drug delivery, and the founding chair of formulations design and development. He serves on the editorial board of Pharmaceutical Technology, International Journal of Pharmaceutics, AAPS PharmSciTech, and Drug Delivery and Translational Research. As a formulations expert, Dr. Khan served as a FDA representative to EMA, PDCO, WHO, USP, NIH, DoD, DARPA, NASA, and the Bill and Melinda Gates Foundation. He has also served as a policy advisor to the CDER Center Director, where he helped resolve complex issues of drug reviews compliance with science. He led the chemistry review team that approved the first 3D printed tablets in August 2015. Dr. Khan's research is supported by NIH, FDA, and pharmaceutical industry researchers. Really happy to have you here, Dr. Khan. If you all would please join me in welcoming Dr. Khan.
Thank you. Thank you very much. Good morning, everybody. I know it's noon there, but it's still morning in Texas. I'm sorry my voice is a little bit low. Hopefully, you'll be able to understand me and hear me. Let me pull my presentation from here. If it's okay, let me share it. Are you able to see this?
Yes, Dr. Khan, we can see.
Very good. So thank you for this opportunity. It's great to be with you all. I'm very passionate about this field. As you can see, it provides a lot of opportunities to our patients. So I'll talk about 3D printing and bioprinting, and I'll provide some examples from our laboratory experiments. I'll talk a little bit about the mechanism and how do you control the quality of this 3D printing and what impact it has in the pharmaceutical discipline. So the outline and the scope of the presentation, so 3D printing, a little bit about bioprinting. I'll talk a little bit about historical development of this one and talk about different 3D printed technologies and mechanisms and how we are utilizing one of those technologies, actually 2 of those technologies that I'll present, and some regulatory considerations of how you can actually implement and provide it to the patients, right? So we'll talk a little bit about it. So the 3D printing, it describes various methods of, so how does it work, right? It describes various methods of construction objects in a layer by layer fashion. That's one of the advantages of presenting. And later on, you will see how you actually control and give precisely the same dose to, especially the pediatric patients every time, right? So it has a layer by layer fashion. An early concept actually came from MIT, and where they had a liquid binder onto a thin product. So you have a powder blend containing the drug and excipients, and then you spray a binder on it to obtain a shape in the form of a tablet, right? Now there are many types of mechanisms, right? But in almost all cases, the object to be printed, for example, you're printing a tablet or an organ or anything, the object to be printed is created using a CAD software design. So that object is created on a computer-aided design, and it is then exported to a file to be printed. And the exported files, it splits that object into various layers. No matter how complex that structure is, when you see layers, it looks very easy when you have a layer, right? So it just prints layer by layer, what's there in that layer, right? So that's how you obtain a 3D printer. So essentially what you need in 3D printer, you have a hardware, which is actually a 3D printer that you see. It looks just like a regular laser printer that you have, right? A normal laser printer that you have. Some of them look just like a regular laser printer, yeah, the benchtop laser printer, right? So it has a 3D printer. The software that I told you, that's where you create that object, right? The CAD design. A lot of engineers and computer scientists, they utilize this, and architects utilize this routinely, the CAD software, right? And then, of course, you need the material that you're using, right? The qualified materials that are that are approvable by FDA, you need to pay attention to that, right? Now, in the bioprinting, the 3D printing has really advanced. Now you can have biocompatible materials. You can have cells, and you can construct some tissues from those cells. And with bioprinting, you have the potential to develop actually desired tissues, and organs can be printed, right? So very specific models, devices, implants, a lot of those things can be printed by 3D printing, right? Just an example here, for example, if you are in a surgery, you're printing, you can see the layers and layers of this structure, the bone structure you can create an object. Just take a picture. I mean, you can have an MRI, and you can have the CT scan, you can have those scans, and then you can layer them and then print through cells and tissues. You can do that in surgery. In disease modeling, I see some of those, my colleagues in Texas Medical Center, they are doing some of the receptor and the disease modeling, right? So they create some objects to understand how the drugs work. And I see some folks actually printing certain organs here, right? So I see in the news all the time, right? Some heart is printed, some ear is printed, as different organs are printed. So you can have this organ printing, and you have dentist. You can actually print different types of, implants, or you can type different types of teeth, things like that, right? But our interest and my background has been in pharmaceutical products. So I'll spend a lot of time on pharmaceutical products. These are tablets here, as you can see, so we can see what we need to print and how we print the tablets, right? So I'll spend some time on product. But historically, just one slide of that, somewhere around 1980s, where Hideo Konama, he has actually printed a 3D printed plastic models, right? And this was in XYZ printer. So the product that FDA approved, the Aprecia formulation in 2015, all the initial studies were done with this XYZ plotter for them. And eventually they used binder jetting for printing the 3D printer product. But just know that the very initial prototype was used where an actual formulation was developed for FDA approval, right? Now, in about 1984, Charles Hull, he used the stereolithographic technology, right? And so, eventually it became the 3D Systems company. And around 87, that's when you have this SLS technology, where you are doing this sintered lasering. So this is from University of Texas, and 89, that's when you have fused deposition modeling, where you will see some example in the coming slide, how they do this fused deposition modeling, where you can get some control release products too with this one, right? You obtain some filaments through an extruder, and from that filament, then you can have a product for sustain and control release also for 3D printing, right? And the last one, 1993, as I said, this is a binder jetting where you are spraying binders on a material to obtain the shape, right? These are different types. So why this 3D printing became very important for us in the laboratory? Because I was looking at some pediatric products. When I was in FDA, I was an FDA liaison to EMA, where there's a PDCO, Pediatric Development Committee. Every 2 weeks, we used to have meetings and we were discussing challenges of developing pediatric products, right? It's not a great profit for the industry. That's why you have BPCA and PRIA, you know, utilizing this carrot and stick model to, entice the companies to make products for pediatrics. And they do make pediatric products now, but still there are a lot of products for which there are no pediatrics products at all. It's very, very difficult to do studies. Now, some of those challenges, why this not very profitable and challenges, is the heterogeneous population for pediatric. A child is 2 months old, 6 months old, 2-year-old, 6-year-old, 8-year-old. They need different doses. And FDA has a product defined by a dose. So you have 2 milligram tablet is a dose. If it's a 5 milligram, that's considered another product. 10 milligram is considered another product, right? And so you have different. So this 3D printing provides flexibility of dose, right? So and several dose calculation approaches for pediatrics you use, if you have a 100 milligram tablet, a child might need only 2 milligram or 3 milligram or 8 milligram. How do you come up with 2 or 3 milligram when you have a 100 milligram tablet, right? So it's a challenge. So people utilize different ways. For example, age in body weight or age in body size or body surface area. So depending on the age, they calculate the dose based on the age, right? That's what they do. But a child is not the same as an adult, right? Because, there are a lot of differences between adult and a child, right? So for example, the gastric emptying time for a child is variable. The gastric pH in the children, they tend to have a slightly higher pH than adults, right? And surface area of the absorptive sites, you can easily imagine a child having very different surface area, permeability. They are not well developed. It could be a whole lot more. The biliary functions are not well developed for a child, right, as opposed to adult. And the body water composition, child may not have a lot of fat, but it has a lot of, the body composition there, right? And the transporter expressions are not well developed. So when you use the age model or when you use the weight model for the child, obviously you won't get the accurate dosing, right, for the child. And so if the studies are not done and if you're calculating the dose based on the adult dose, obviously children will not get the right medications, right? This is a fundamental problem in pediatric dosing. Now, I had heard this slide from Ann Zajic from National Institute, from NIH, or when she was in NICHD at the time. And I think since she has retired since then, but this slide was very valuable to me because Anne was presenting that, hey, we want a product where you want safe excipients and you want very stable products, stability under, because, if you have a liquid formulation and it's going through transportation and other issues there, you may have excursions of temperature which might make the product unusable, right? In some cases, or you may have fungal and microbial growth and things like that, right? Uh, Palatable. This is a very important point to understand. If a drug is very bitter in taste, for you and I, for an adult, what they do, they provide a coating to a tablet. So when you take a tablet, it's coated, you don't feel the taste. The drug might be very bitter, but because of coating, you don't feel the taste. But if you have the drug crushed and ground and made a liquid out of it, all the taste is exposed, right? So that's a huge problem for children, palatability. And sometimes they cannot swallow the tablets. And you need an ability to titrate doses. That's where the dose flexibility comes, adequate bioavailability, and avoidance of extemporaneous compounding. This is an important point. I mean, I'll bring it in coming slides. So all these things, uh, how do we accomplish all these things, right? So that was getting me to think about when I was in FDA itself, how to come up with certain strategies for development, right? And so, like I mentioned, this is State Board of Pharmacy, they published a paper. They did a thorough evaluation of compounded products, and they looked at products between 2008 and 2010. This is what they found. They found that there were palatability issues, dose accuracy issues, and quality. The basic potency test for the compounded product, right? The products that are compounded given to pediatrics, most of them were failing, right? Potency range was from 59 to 89%. The allowed potency range for approvable products usually is between 90 to 110. And in some products like levothyroxine, it's 95 to 105. That's how accurate you need to be. But you see, their accuracy is 59 to 89%, right? Most of them, a lot of products were failing. A lot of them had stability issues so they published this paper with a very thorough analysis of a lot of compounded products, right? So this is a slide I use just to demonstrate, on the left side, you're seeing children, they're growing, and adults. Once they reach an adulthood, pretty much their dose is constant. On the right-hand side, you're seeing their dose is constant. So for example, you have a 25 milligram tablet. So we all take 25 milligram tablets, we're happy. But now that 25 milligram tablet, what this pharmacist is doing, by the way, I started my career as a pharmacist. My initial job was in New York City, and I was literally doing this. I was literally compounding products for pediatrics as well as some of the people. Whenever the strength is not available, I would take an adult tablet or a capsule. I crush it and grind it and put some OraSweet or NutraSweet, some kind of a liquid there in it. So the solid tablet, I made it into a liquid formulation. That's what most pharmacists do even till today, right? And just for bringing the dose for these pediatric patients. But again, that brings stability, bioavailability, quality, potency, taste, uh, sterility, the fungal and microbial growth, all those issues come about in compounding, right? So that's why we thought, won't it be great to have a 3D printing where you're actually layering drug and you can easily control the amount. You need 3 milligram, you have certain number of layers. You have 8 milligram, you have certain number of layers. And we proposed this, NIH. I'm really grateful that, they understood that. And the reviewers have liked the proposal. We got a couple of, actually 2 R01s and 1 R56 funded. Just now I got a grant from NIST. And then the Texas state is funding. So it is increasing. And now, but the initial fundamental help that we got was from NIH to help us progress, and we continue to work on that one. Now, just to see the technology a little bit, you have fused deposition modeling, you have stereolithography. The last two are the ones we actually work in the laboratory. And in the binder jetting one, where I said you have a binder, you just spray it on a powder, right? So liquid binder is sprayed on this material, right? Then you have stereolithography. You have a container filled with a photopolymer, and that light is projected on that, and so it takes some shape of that one. I don't normally use it, but it's very good for certain other objects or organ objects, right? Now, selective laser sintering, as I mentioned before, this is a powder bed fusion here in this one where you have powder, but in the powder we add a new excipient. Now, by the way, whenever I say new excipient, whenever you add a new excipient in formulation, FDA has a lot of studies that we need to demonstrate. We need to do mostly tox studies, right? So by new, I mean an existing excipient which FDA approved, but that excipient has a capability of sintering the material. What it does, that excipient melts with laser. I add a new excipient in tablet, that excipient melts and it gives the shape and form for the tablet, right? So that's how we are calling it new, but it's already an existing. Mostly these are colors that FDA allows us to use. But if you use a brand new material, you have to do a lot of studies on that one. So, and then the fused deposition modeling. Here we are obtaining like extruded, like, you have spaghettis and, you have extruded material. So you have your tablet material first extruded and use that as a filament for 3D printing to give a shape, and you can get sustain and control release preparation here on this one. This just shows the stereolithography here. So this is a container in that you have this, material there, and that when some light is impinged on it, these are actually some polymers there on it, right? So, and this is usually good for some hard objects like this. I don't use it personally, but I see some other people are using this material. Now, the advantages, you can get high resolution printed products, you can print products, right? But biocompatibility might be an issue, and the photoinitiators and that are utilized for making the polymers, they may have some residual toxicity. They may be very cytotoxic, right? So it requires some extensive work on that one. Fuse deposition modeling, as I indicated to you, so you have the filament that's extruded from this one, and that filament you're using, this is very common, using filament or something to make a 3D printed object. These days, I see even high school students doing that. They have some filament plastic material, they make 3D printed objects. It's very easy. But the advantage of that is you can have a delayed release, you can have immediate release, you can have control release, you can, because of different filaments that you can obtain, you can have different types of filaments to get. A filament can give you this drug A, another filament can give you this, and a third filament can give you this. So you can have a combination of layers there and control release products there on it. The problem with this fuse deposition is you need high temperatures for it. If a material is thermolabile, then it's very difficult to utilize this technology. Now this, as I mentioned, it's very amenable to the kind of products we are making, very easy to use. It's solvent-free. You're not using any solvent. All you have is a powdered material, drug and excipients, and then apply some laser to it, and laser will sinter the sintering material in it, giving it the shape on it. So it's very, very practical to use. And so you can get printlets of different amounts and different types. Now, one of the neat thing we are exploring with this one is to have a combination product. And I think the sky is the limit for it. Hopefully somebody from the audience will come up with 3D printed. Sometimes I see some family members using 8, 9, 10 medications, or 7 or 8. Or can you imagine 8, 9, 10 medication in the morning, another 8, 9, 10 medication in the evening, and then it's very tedious, very difficult for a patient, especially elderly patient, to have all those drugs. They remember all those drugs and use them. Now, theoretically, is it not possible to have those 7 or 8 drugs and make a one pill that immediately dissolves or disintegrates and elderly person can take? Just instead of giving 8, 9 pills, just give one pill, specially tailored to that patient with the amount of drugs that they need, right? It's very, very possible, but hopefully some younger ones will take that. We have added 2 or 3 drugs, even 3 drugs we have added, but I'm almost sure that we can add even more drugs on it, right? So in this one, so this is an illustration of that one. So this is a powder reservoir, but where you are making the tablet through the CAD design, you have a spherical shape here. So laser will go in a spherical shape here in this one, and it forms, even though you have a powder all over, so it forms the tablet in the areas that you want, right? And you can get different shapes to it. One of the challenges when we were sending papers for publications, right? Some of the questions they asked, and also the sponsor, when they submitted this product to FDA, the issue was, yeah, you got the tablets. What are you doing to rest of the powder? So what we do, we recycle that powder. So one of the objection was you're already exposing to a lot of environmental conditions to this powder, but you are resending this powder. So can you recycle this powder? And how many times you can recycle the powder? And will the tablet change if you recycle it, right? So those are some questions that needed to be addressed, and we've done some work on that one. So the printer looks something like this, just looks like a regular 3D printer, a very small device that you can keep it on your table, just like a laser printer. Looks like this, but inside there's a chamber here. That's where the tablets are getting printed here. But when we're using SLS, it's very important that we understand the interplay of powder bed temperature. What happens if you increase the bed temperature or increase the laser speed or formulation, the drug that you're using, the particle size, the density, because your goal is to provide accurate amounts to the patient all the time. Otherwise, you saw Anne's slide, right? The potency and the quality of the medication is important. If we understand the interplay of these factors, then we can produce tablets that are consistent over and over again, right? And one of the ways we understand different factors is by utilizing the quality by design principle. FDA have guidances, EMA has guidances there, and there are a lot of ICH guidances, Q8, Q9, and Q10. And what the guidance tell us is look at all the variables. For example, the sinitering material that you're using. What can go wrong in the sinitering material? Now, this is not a experimental exercise. This is a table exercise where experts sit and think what can go wrong, right? So they look at all the possibility of changes, sinitering parameters, the layer itself, the environmental factors. So you look at each one of the factors and you calculate a number called R.P and risk prioritization number, where you see a severity, occurrence, and detectability of each one of those factors. Now, if you want to do experiments on all these factors, there'll be a lot of experiments. You might have to provide hundreds of experiments. To cut down on the number of experiments, you do the risk prioritization numbers. If the risk is very high, then what you do, there are certain factors where the risk is very high. You select a designed approach. If there are still many factors, then you can use this design. If there are a few factors, you can use this design, right? So you can approach different types of a design, right? And for example, if I want three factors with three level of interactions, I use a full factorial design. So I understand the project, I understand the variables as well as their interactions thoroughly if I use a full factor, but that will have a lot of experiments I need to perform. But if I need to perform just a few experiments, I can do a screening design. You see, I just have only on the edges of this box to provide to do only a few experiments, right? So you select the nature of the design depending on the issue that you have and depending on the material availability. Now I'm providing an example of lopinavir that we wanted to just try out some experiments with. And so we have prepared some different formulations of lopinavir 1, 2, 3, 4, and each formulation has different compositions in it, right? And we obtain these tablets, lopinavir, see different types of 3D printed product here, right? And again, these are initial studies where we were trying to see the texture here because after we make the tablet, they should be readily dispersible or readily dissolvable. If a child is taking it, you put it in a spoon, the tablet, a little bit of water, it just goes into solution right away, right? And they are very stable, right? Now, and there are different variants of that one. Now what we are trying is we are trying to first coat the material and then 3D print it, right? There are different ways. But the important thing is if the micro texture you can see, this will be easily dissolvable as opposed to these two. These are amorphous here because we notice that if a material is crystalline, it doesn't dissolve much faster, but it's amorphous, it dissolves right away. Or sometimes you have a fraction of the amorphous and the crystalline material to control how much of the drug dissolves, right? And so these are different formulations here. I'm trying to remove this here. Okay. Now, when you do a design set of study, you can actually predict that if I have certain excipients and certain compositions, what kind of products we get, how much crystallinity that you will have on that one, right? So these are some of the predictions that we had made by the design set of studies, and the actual crystalline and amorphous material, as you can see, it was very, very close to the predicted ones, right? So this, we have published this paper. You can see more details on that one. Uh, and how do they dissolve and how do they dissolve after 1 month? So you can see when we have certain formulations after 1 month, we saw that some of the amorphous material was getting converted back to crystalline material. So if I see this formulation, lopinavir, this formulation 1, you can see it was dropping, right? Now this one was not dropping. This one was not dropping. So it was giving us an indication of stability of this product, right? Now I will talk a little bit about pyrimethamine tablet. This was actually very interesting because this was for use for toxoplasmosis. I'm not sure if you all know about it, but there's this company, the Viera, they had this pyrimethamine tablets, $11 or $12 medication became $750 because the market competition wasn't there. And so this was a case, FTC, Federal Trade Commission has gone after Viera. So, but I realized at that time um, that the 25 milligram tablet was $750, and that toxoplasmosis is a parasitic disease was for adults. But what about children? $750 for an adult pill, and if I need, it's a 25 milligram tablet, right? But if I need only 2 or 3 milligram for a child, I still have to pay $750 an adult pill for that one, right? So that's what we propose for toxoplasmosis. Can't be 3D print 1 or 2 or 3 milligram, right? And that's why this proposal got funded. And so we obtained some pyrimethamine tablets just to give you an idea. This is, if you see the tablet from top, it looks like this. From the bottom, it looks like this. So if there is any difference, that you will notice the difference on this F3 here, right? So we can do this chemical imaging of that one. This shows the amount of drug dissolved with different pyrimethamine printlets that we obtained. We can get the amount that we want and the release profile that you want, right? So we were trying to figure out what kind of release profile you get with these pyrimethamine tablets, right? And after we obtained the tablets, we even did a bioavailability test or a bioequivalence test by commercially available pyrimethamine tablets. We took a commercially available pyrimethamine tablet and we 3D printed the tablet. Our goal was to obtain similar kind of profile as the commercial tablet, but this is available as 3D printed, right? That's what we did. So this was done in some animal model here, and we saw the plasma concentration here. It's fairly closely mimicking the compressed tablets. We saw some of the anti-tox plasma activity here, and where we saw luminescence readings here. So these are DMSO controls for that activity. As you can see, Toxoplasma gondii, these organisms, they were not going down. But when we use pyrimethamine, the red ones, the luminescence assay value was very, very low. So it was very effective against Toxoplasma gondii. And then when we use the tablets and our printed, they were also equally effective on this one, right? So we had to do some efficacy study by seeing the multiplicity of infection of Toxoplasma gondii, right? Now, I mentioned to you about the recycling of powder, the issue that we have for the recycling of powder. That's why we had to do some studies by taking the recycled powder. This will show you if we recycle it 1 time, and then we did assay. If we recycled it the 2 times, we did the assay. We recycled it 3 times, we did the assay, and then we did the dissolution, we recycled. And so by this way, this was for, so when the question was asked, when we sent, the reviewer had asked this question. Luckily, we had this data for another phenytoin that we had obtained. So even though we did not have for pyrimethamine at the time, but we sent this data and we convinced the reviewers that with recycling it 3 times, it's not an issue. But we noticed that after 6 or 7 times, the quality was changing dramatically, right? But after this, we did do the recycling study with our products, also pyrimethamine, and it was very consistent. Yeah, this is for pyrimethamine data, so the recycled data that we have here. We obtained a lot of formulation pre and post, and we had pretty consistent results. In binder jetting, we are still working on it, as you can see. So this is one drug, this is the other drug, right? So we're trying to combine these two because they don't stick. There are a lot of parameters that we have to play around. This is just the initial reading at this time. We are able to get these bilayer tablets, but we will try to increase the size of that one so that they are not very thick. But by principle, it is working. So now I will talk a little bit about, I mentioned to you when we do the screening study in the different types of experimental design. And so, for example, this is formulation number one, this is lamivudine. So these are 5 factors that we have here, related to spray, related to thickness of the film, of the thickness of the tablet, things like that, right? So the negative sign here means you have that factor at a low amount. A positive means you have that factor at a high amount. These are 5 factors on that. So each product was made with different patterns, different experimental patterns here, right? And 0, 0 is the mid-level of those factors, right? And it's just to give you an idea. So the dissolution will be very different. You just have to select what we want, right? So you can get different types of release profiles. Now I have this picture to demonstrate. This is something for uh, uh, this is powder here, and you spray the liquid here on this one, right? So you have a layer of powder that comes, then you spray. Then you have another layer of powder that comes, then you have spray. Then you have another layer of powder, then it sprays again, right? That's how the tablets are made, and that's how they are approved. Now, the defects of tablets, this is a totally new area, right? Because in normally when people make tablets, they have capping and lamination and mottling and those kind of issues they have in tablet. But if you have 3D printed tablets, their their defects are very different. You have something called banding here. There you have the sides ripples on part sides in the XY plane during printing you will see, right? You will see leaning, you will see warping, you have see stringing. So a lot of new terminology we have to get used to in pharmaceutics, but those in 3D printing area, they know this very well. By the way, when we did this 3D printing, what we did, we collaborated with engineering folks on this because they know 3D printing very well. They know the CAD design. And so it has really cut down our time. Our collaborator is from College of Engineering here. That's why it made it a lot faster for us. And this is the paper from FDA that shows some of the regulatory issues that we faced when we published this paper. Now I'll tell you something very important. Now each tablet that we prepare, every time that we prepare tablet, we have to do a lot of characterization work. I told you about some of the X-ray diffraction here, the Bruker, and because I told you some of the material is crystalline, some of it is amorphous in nature. You look at particle size, the DSC, the TGA. You need all these instruments here, and then when the product is made, this is the material characterization on the left-hand side, and on the right-hand side you are seeing some disintegration tests, you're seeing some dissolution tests, another type of dissolution test. You're looking at some of the porosity, you're looking at friability, you are looking at from the tail vein some of the PK work. By the way, the PK work we did on a rodent model, and then we did a non-rodent model also. We did it on beagle dogs as well, right? And you're doing chemical imaging here. Now, we want to deploy this 3D printer in a hospital. That's the goal, right? That's what we are heading. But most of the hospitals do not have that. Most of the pharmacies do not have all these technologies, right? So how do you ensure the quality? That's a very important task, right? And that's why I wanted to have engineering and pharmacy students work together to come up with a strategy for that one. We are very pleased that we have come up with. By the way, this is not published anywhere. Our first paper just got accepted last week. Nobody has ever done that. Nobody ever published that. You will not see a single paper on the real-time monitoring of 3D printing where I'll show you what we did. So this is the 3D printer. This is the, and thanks to NIH for helping us with this. This is the 3D printer, right? But so once you obtain these 3D printed tablets, they need to be subjected to all the tests that you saw in the previous slide, which hospitals do not have. Those big laboratories with mass specs and HPLCs and UPLCs and DSC and TG and all that, they don't have all that, right? So I challenged our students and postdocs to come up with a system. Now they have this system here that they developed. By the way, they got parts from Amazon and other places. They have merged all these parts to come up with a system because nobody makes such a system for 3D printers, right? Now we have done a lot of work for other systems for mixing, granulation, or for 3D printer, there was nothing. So they actually physically drilled a hole on the 3D printer. They mounted these things, and so the probe goes here on the 3D printer after every layer is printed, after every layer is installed on the tablet. This will go and give a scan on the left-hand side you're seeing, right? You're seeing a scan like this. So if you print the tablet, usually in about 20 to 30 minutes, you have all the tablets printed, like 30 to 40 tablets you print, right? By the time a patient comes from a physician to a pharmacy, his tablets are already printed. Any new tablet is already printed, right? So that printing, as they're printing for every layer, you get a scan like that. And now I had some chemometrician and some big data analytics folks. They work on it. See, all the data is there on these graphs, and and so all the physical changes and all the chemical changes, they're able to identify those things, right? And so this is this is the probe of that near IR that it's taking a picture like this. Now, if you do that, you can see all these properties. They go in those scans, and the scans will look like this. Regardless of what you take, after every layer, you're getting a scan, right? So if I print this for 30 minutes, this is the data I have in 30 minutes. I have all the information here. Now, the chemical peaks will tell me if there's any chemical change, if the amount of drug is getting added or coating is getting, whatever you're doing, right? So it's getting added. And if there are any physical changes, the tablet is getting deeper and deeper, the slopes increases, right? So we have to, if this data, we have to pre-process the data, right? So if I don't have a lot of useful information on the left side, I can truncate this. If I don't have useful information, I can truncate this so that I'll have less usage of my space, right? So this is the truncated data, some of the truncated data. And if there's a lot of scattering here, the instrument corrects for multiplicative scattering here for that data. But the bottom line is, after the corrections and pre-processing, we take a derivative. What the derivative does for us is it will clean this area so we can, just like an HPLC, we are using the peak heights versus the amount. In this particular instance, this is a coding peak versus uh, versus uh, the peak intensity. So just like that, you can take the tablet depth versus intensity, or the drug amount versus intensity, or dissolution versus intensity. So you can have different peaks you can utilize for different purpose here on this one, right? So this is a work in progress, but the preliminary work that we did, some of the preliminary work we did, we've already published on that one. But and then by utilizing that peak, you can see the actual versus the predicted values of them. So I will conclude that 3D printing may provide a paradigm shift for certain low volume product. By the way, it's not very good for very high volume product. That's why we selected like toxoplasmosis. You don't have a lot of patients. By the way, I saw some malaria cases in the United States recently. There was one case in Dallas and 4 or 5 cases in Florida. So, and you know how many people make drugs for malaria here? There are not many patients here. So this 3D printing is very, very good for them. So whenever a situation like that comes, or if there's a drug in shortage, situation like that comes, you can easily 3D print those medications for those patients. So it may provide a paradigm shift for certain low volume products. case need to be paid for marketability. And see, nomenclature is also very important here on this one. What do you call them? If you call them a tablet and somebody else is also making a tablet, which is much cheaper, then why do they do this 3D printing, right? So, so that's something we need to work on with FDA for that one. The Aprecia did not get a new nomenclature. They got a tablet, 3D printed tablet. Now we can have a personalized on-demand. Can you imagine for cancer patients and other patients who need a flexibility or dose changes? A lot of narrotherapeutic index drugs where they need to change the dose for a patient, right? So 3D printing is very, very good. Enhanced stability because you are completely preventing solvent. A lot of times solvents, that's the one that cause oxidation, hydrolysis, and things like that for ingredients, right? So you're preventing all that. And by the way, with dead stability, even for 6 months at 4075, they're fairly stable products, right? The risk-based production is feasible. That's in line with FDA's quality by design. Technological innovations are needed. So one of the challenges is the yield of the compound, right? So because you saw the material, you have to recycle, so you might lose some compound there, right? So with practice and experience, the yield can increase, right? But you can make immediate release product, you can make extended release product, you can make delayed release product, especially you saw the fused deposition modeling, right? It's very amenable, right? And bioprinting too. I haven't covered a whole lot of bioprinting, but some of my colleagues are working here in Texas Medical Center. They've done a lot of work in this area, which is very fascinating. And so I want to thank NIH again for the funding they provided. And this PhD engineering student now, he's graduated now. And some of these folks are going back and working in FDA, and some of them are working in the industry now. A lot of postdocs and students got trained here, but everybody had a job, and everybody is very decently employed outside. So with this, I will stop here and thank you very much. Any questions, I'm happy to address. Thank you very much.
Thank you so much, Dr. Khan. This was a really insightful, engaging presentation. You just your explanations were clear and thorough. Really appreciate your thoughtful perspectives. I think overall it just made this topic, both accessible but also inspiring too. So, just thank you for your presentation today.
Thank you very much.
So I think we can just jump on in with a couple of questions. To our audience, if you have any follow-up questions, please feel free to put them in our Q&A box. And we will make sure, or at least attempt to get it to as many questions as we can. So perhaps we can start with one of my questions, Dr. Khan. You mentioned sort of like that 3D printing, could likely solve compounding problems for children. I guess I wanted to know, like, what do you think is like the biggest, maybe technical or regulatory hurdle to ensuring that every 3D printed pill meets the same safety standards as a mass-produced one?
So the few that we have done, we have actually compared head to head with the mass-produced one. We have come to tell remarkably, because actually it gives you flexibility. Like I said, you can have increase or decrease resolution. You can have increase or decrease of hardness and friability, but you can select the ones that are matching the commercial one. But a lot of time when you're doing compounding, for example, if you have a tablet and you crushed it and you ground it and you made a suspension of that tablet, and that's what a pediatric getting. And what's the quality of that medication? Who's testing the amount of that compounded product? Nobody's testing, right? Pharmacists do not do that. I used to work as a pharmacist myself, right? I crush it and I grind it to make a suspension and give it to the patient. I think it is 3 milligram by calculating the aliquot, but I never know because I never tested it. We don't have equipment. Here you actually have it tested. You have all the properties tested. Now, if it is for commercial production, then it has to go through FDA approval process, just like the Aprecia and that formulation did. But if it's compounded one, we don't need that. You don't need to have those evaluations at all, right? But we do that. We do that and we publish it routinely. And there is no chance of drug getting degraded there in the conditions that we are doing there, right? So, but we have not only physical chemical measurement, we also have the bioavailability and bioequivalence measurements in 2 animal species. So it's a whole lot more than what the pediatrics get with their compounded product. So if it's
Wonderful.
compounded, you don't need FDA approval for that one. It's controlled by state.
That's an, that makes life a little bit simpler, right?
That's the advantage. That's the beauty of that one. So in a compounding practice, you have a physician and a pharmacist and a patient relationship, right? And physician knows if the drug is not getting adequately, there's a problem, then it can be corrected right then and there itself. It doesn't happen. But by the way, what I did not present, Dr. Samari Bates, is that we have an electronic tongue, and that electronic tongue tells us the taste of this one. It's very useful. So we have a human panel taste study we're doing right now. We already have the electronic tongue data. We're trying to match the human panel taste testing versus electronic. So you get a good handle on the taste and the stability and the bioavailability and the potency. So you're getting all that which you don't get the compounded product.
Wonderful. Thank you, Dr. Khan. Great response. I guess I also, you were talking about, like the clinical settings and the clinical and the pharmacy. So maybe a shameless plug for pharmacists, but, maybe do you envision maybe a world where, the hospital pharmacy or even the neighborhood pharmacy maybe kind of evolves into like a mini manufacturing site with its own 3D printers? Do you think that that could be something?
It is, our partners are Driscoll Children's Hospital. That's the proposal we have, and we are working with them, and we are trying to have a clinical pharmacist working with joint funded position between Driscoll Children's Hospital and us. And this pharmacist will be trained in that. It will actually be deployed in the hospital. In the next 2 years, you will see that. You will see there's a very good chance that it will be actually deployed there. And they because for them, it's a whole lot better than a compounded product. And you
Mhm.
can get the taste that you want, you can get the flavor that you want, you can get the shape that you want, you can get the amount that you want, those flexibility, and it's printed right then and there itself, right? So they also see a lot of value in it. And by the way, I got a call from UT San Antonio Health System, that they are also very interested in that one. And we are trying to establish collaboration with other children's hospitals, but it's very practical, Dr. Bates, for compounding.
Wonderful. That's pretty exciting, right? To even be talking
That's
about something like that. That's wonderful. Um, so it looks like we have a question in our Q&A. This is an extremely exciting area. Thanks for the thorough, informative presentation. Do you envision that the real-time in-process monitoring tool could become integral component for quality control documentation, for the pharmacist before dispensing the 3D printed pill?
It requires a lot of understanding and technology, the bit trader treatment and all. Hopefully, with time, it will eventually become because they see the value in it, right? If you don't do that, for example, you're thinking it is 3 milligram, you're thinking it's 5 milligram. Compounding products also, they think it's 3 and 5 milligram, but they're not sure about it. So you always have skeptics of that, right? So you may have skeptics of 3D print. So it's to our advantage to use it. Now, the thing is, it's not commercially available yet, and we're trying to patent it. We're working on it now. We're trying to patent that. And so hopefully some other developer will take this and commercialize it.
Fantastic, fantastic. Um, I think we might have time for one more question. Um, what opportunities and potential limitations on bringing 3D printed medication tools to low resource settings?
So if you see the overall resource that is needed for this as compared to the development, because as I had indicated to you, you don't need a mass spec, you don't need HPLC, you don't need UPLC, you don't need any one of those things, right? So, and the 3D printers are not very expensive. It's just like your laser printer. Actually the very first one we got for about $10,000. I know it is an expense, but if you compare the expense of all the development that goes out there, that goes in millions, that
Mhm.
equipment, right? So it's a whole lot less than the commercial ones, right? And so once you start doing it, you'll realize it's a very profitable to do that. It should be very profitable because it's not very expensive at all.
Great. The, Dr. Khan, I'm receiving some messages, about how this is a great presentation, and thank you. I think you've got the gears going. You have people that maybe have never thought about 3D printing in this area, and maybe this, you know, will sort of expand the field, right? I see the field only growing exponentially. So really, thank you for maybe sparking that innovation and that uh, the creativity in our audience. So thank you again. This has been a fantastic presentation, even better Q&A. So thank you so much for
Thank
being
you
here
very much.
today.
Thank you very much. Thank you all very much.
Wonderful. So I'd like to go ahead and close for today. Just a reminder, our next session is going to be April 8th. So we're skipping a week. So April 8th, our presenter will be Dr. Holly Ruiza of Brigham and Williams Hospital. Her talk is entitled Optimizing TB HIV Treatment in Pediatric Patients. So thank you to our audience, and thank you again to our wonderful speaker. I hope you all have a great rest of your day.
Thank you all very much.